Welcome to part 3 of the Weeks 3 and 4 lecture series. In this portion of the lectures, we're going to delve into what is known as the evolutionary synthesis. We're going to talk about population genetics, and we're going to get into the mechanics of inheritance. And by that, I mean we're going to talk about what DNA is and what it does. You'll see on this next slide here that you're going to have to understand the 3 primary levels of genetic organization. And you're also going to have to understand the process of protein synthesis. And that's basically how DNA translates into the production of the physical body. The work of Mendel that you learned about in the previous lecture laid the foundation for our present understanding of genetics. Beginning in the 1900s, Darwin and Mendel's work was combined, ultimately leading to the development of the field of population genetics. Evolutionary synthesis refers to this combination of insights. Evolutionary synthesis allowed new questions to be asked, and today these questions are answered through population genetics. Once genes were discovered, a new way to measure and track evolutionary processes was born. This is reflected in the definition of evolution above, which is change in allele frequency over time. And we're going to talk more about alleles shortly, but alleles are basically different variants of the same gene. So here we have a map of genetic diversity around the world, which can be measured by looking at the geographic distribution of particular gene variants, which again are also known as alleles. Before we delve into the relationship between genetics and evolutionary change, let's quickly review the basic mechanics of inheritance. So, sections of DNA with specific instructions are genes. Gene variants are called alleles. Chromosomes are organized packages of DNA bearing genes along their length. And a genome is an entire set of DNA instructions inside a cell. So all of your cells, virtually all of your cells, contain your genome. How does DNA sequence impact phenotype? This is a question that we're going to be looking into and that geneticists really focus on. Phenotype refers to the physical manifestation of the DNA. So pheno means to show and typos means type. Your genotype is your DNA. Your phenotype is the collection of all of the traits that your DNA produces. DNA is the primary hereditary material of life. The universally recognizable double helix structure of DNA, which is tightly packaged into chromosomes, was discovered in the early 1950s. It has been described as a twisted ladder with each rung created by 2 linked base pairs, adenine and thymine, or guanine and cytosine. Each of these linked base pairs forms a nucleotide when combined with a sugar phosphate group. The sides of the DNA ladder are sugar phosphate backbones. Sections of this ladder, or particular sequences of base pairs, form genes. There are 3 billion nucleotides, or half rungs, in human DNA. If we were to place all of the DNA from the human body end to end, uncoiled and unpackaged, it would form a microscopic string between 90 to 135 trillion meters long, or twice the diameter of the solar system. The structure and packaging of DNA into chromosomes allow long strands to fit into tiny cells. In each of your cells, 5 to 6 feet of DNA are squished into the nucleus. Chromosomes act as containers to store and protect DNA. Here's a pop quiz. How many chromosomes do you have? If you answered 23 pairs or 46 total chromosomes, you are correct. Chromosomes come in pairs because we receive one from mom and one from dad. Interestingly, chimpanzees and gorillas, our closest living relatives, have 24 base pairs. Millions of years ago, fusion of chromosomes in the common ancestor between chimpanzees and modern humans led to the reduced number of chromosomes in humans. Chromosomes actually fuse fairly often, an error that occurs during the process of reproduction, which may or may not lead to changes in an organism's phenotype. Interestingly, the amount of DNA an organism has doesn't seem to logically relate to its size or complexity. The genomes of our native Californian redwood trees were recently sequenced, revealing vast numbers of base pairs, 9 times more than humans for the coast redwood. Some of these base pairs do not have any apparent function. That is, they don't seem to code for anything that we can detect. Curiously, the genome of the human and the giant redwood tree is dwarfed by the tiny axolotl, an amphibian native to Mexico that can regenerate lost or damaged body parts. Before I tell you about the discovery of the double helix, I'm going to throw in a little extra credit for those of you who are still paying attention to this lecture. DNA and some associated proteins within the nucleus were first isolated, not necessarily seen, but simply isolated in a cell, by a guy named Meischler. Meischler is Meischler in 1869. Meischler took pus off of Band-Aids he collected from people with infections. Meischler, pus, Band-Aids, 1869, DNA. Those are the key points, the key answers to an extra credit question about who first isolated DNA in cells and how. All right, so the first people to visualize or actually be able to see and identify the structure of DNA were Watson and Crick in 1952, working with imagery taken from the lab of Rosalind Franklin. We've probably all heard of Watson and Crick, but few people learn about Dr. Rosalind Franklin, who died an early death at age 37, possibly due to exposure to radiation and a genetic predisposition to cancer due to her Ashkenazi Jewish ancestry, which we've studied in this class. Franklin used X-ray to create pictures of things like DNA before folks realized that X-ray could be damaging to the DNA of X-ray technicians themselves. So Rosalind Franklin was actually an X-ray crystallographer. There are 3 primary levels of genetic information with different roles. DNA copies itself and can be transmitted to offspring. It also produces RNA. RNA acts as an intermediate between DNA and the production of a protein, instructions for which are encoded in DNA. Unlike DNA, RNA can exit the nucleus and travel into the cell to interact with other organelles. DNA must remain inside the nucleus for protection. DNA provides a specific code for the production of proteins. Protein makes up 20% of your body. At least 10,000 different proteins make you what you are and keep you that way. I would like for you to understand the process of protein synthesis. In other words, how does a tiny chunk of nucleotides build your red blood cells, your hair, your skin, or the tissues that form your liver? In the next slide, we'll watch a short video illustrating the process of protein synthesis. I think about protein synthesis like the production of a movie. If you can fill in the blanks on this slide after watching the video in the next slide, you understand protein synthesis. In my analogy, the protein is the film, the final product. See if you can figure out which of the above biological elements could be represented by the necessary crew members and other components of film production.
What you are about to see is DNA's most extraordinary secret. the innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body.
It's a bit like taking the information off a hard disk on a computer and put it into memory, making it a real program that's running. So that process of DNA to RNA is like running a program, double clicking on an icon.
When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin.
All right, so let's take a minute to review and break down the process that you just watched. So here's a summary slide which will hopefully help you pull it all together. Can you use the film production analogy to summarize what you've learned? Now, I'm just going to tell you right now that for some people, the film production analogy might not be super helpful, but for me, it is. And oftentimes for concepts in biology, it really helps to create some kind of a scheme that helps you to memorize a concept. So here is my memorization scheme for protein synthesis. Number 1, there are 2 phases. Phase 1 is called transcription. This is really important. This is going to show up on your quiz. So during transcription, mRNA copies DNA. Second phase is translation. And during translation, the mRNA leaves the nucleus. Recall that DNA cannot travel outside of the nucleus. The nucleus is there to protect the DNA. That's why RNA is so important in part. So messenger RNA leaves the nucleus with its little copy of the DNA, and it works with transfer RNA in the ribosome to string together the amino acids, which creates a protein. A string of amino acids creates a protein. So here we are with our film analogy. The movie itself, or the film, is the protein. The DNA is the script. The actors are the amino acids. The director is the messenger RNA, who's basically telling everybody what to do, more or less, based on instructions from the DNA. Then we have the transfer RNA. And if you think about it, the transfer RNA was represented by those little green structures with the red tips on the end, and the red tips were the amino acids. So in my mind, transfer RNA is a bit like a casting director who finds the specific actors for the proper roles in the film. And then lastly, we have the actual set of the film, where the film takes place, and that is inside the ribosome. So where is protein actually produced? It's happening inside the ribosome. And this concludes this particular portion of the lectures, part 3.